Um, so welcome everyone to our second seminar of the COVID seminar series. It's my great honor to introduce Dr. Brigitte Gomperts. I'm sure a lot of you know Brigitte. She got her MD from University of Witwatersrand in medicine and surgery. And she came to UCLA, set up her lab as an assistant professor in 2004, and she quickly rose through the ranks to full professor within a few years. And she was also selected to be the co-director of the JCCC Cancer Stem Cell Biology program in 2014. Brigitte has received many prestigious awards. She was the elected fellow of the American Academy of Pediatrics. She received the AFMR Pulmonary and Critical Care Scholar Award, and this very prestigious pediatrics science award, the Harvey Colton Award by WashU. She's also given lots of seminars on lung stem cell therapies and regenerative medicine at CERM workshops, and also National Academies of Sciences, Engineering, and Medicine. And her lab studies lung repair and regeneration, specifically mechanisms of normal repair and how they are altered in different lung diseases. And so we're all very, very fortunate to have her as a great colleague and collaborator, because you can imagine how well sought after her 3D lung organoid model is could be applicable for the COVID research currently. And I don't know how she managed all these collaborations, because everyone wants a piece of her. And I think she would tell us about this amazing 3D culture system where it actually provides a relevant primary cell model to study the interactions of different cell types with the virus in a more relevant lung microenvironment. And so it's my honor, and please take it away, Brigitte.
Well, thank you so much, Melody. Thank you for such a kind introduction, and I'm really happy to be able to talk to everybody today to tell you a little bit about what we've been doing with really repurposing the lung models that we had in the lab to study other diseases for COVID-19 work. And so the way I thought I would break this down today is to first of all give you a little bit of background about the lungs and the structure and function of the lungs, so we're all on the same page. I want to talk a little bit about the pulmonary complications of SARS-CoV-2 and COVID-19, the disease, and then move on to the models. And then I also wanted to share with you some preliminary data. This is all unpublished data, and I'd love to get people's input on what you think about the data, and also hopefully this spurs more collaborations from people. We're collaborating with lots of groups, but we're super happy to collaborate with more groups as well, because we think obviously this is a really important problem, and we're hoping our models will be able to help find some therapies for the disease. So let me see if I can advance my slides and get a pointer up as well. Okay, perfect. So the lungs are actually very complex organs, and obviously are vital for life. And they really extend all the way from the upper portions of the lung, which is really the mouth and the nose and the throat, down through the trachea and the bronchi, which are these cartilaginous airways, down through the bronchioles, and then down here, the bronchioles here blown up, and then down into these little alveolar sacs. And each of these regions is highly specific and different, and has a different function. So as you can imagine, these upper airways, as well as the trachea and the bronchi, are directly in contact with the environment, and so they have a very important role in host defense. So for all the bad things that we breathe in, like viruses, bacteria, pollution, we have a very efficient mechanism of clearing these bad things. And through the way we do this is by generating mucus. And mucus traps these bad particles, and then we have ciliated cells, which have these hair-like projections that beat unilaterally, creating what we call the mucociliary escalator. which allows the mucus to be moved up and out of the body, so either swallowed then down the esophagus or coughed up, and in that way we clear all of the bad things that we're breathing in. And then if we move down to the alveolar sacs, there's a very different function here, because here we have these very, very thin air spaces surrounded by cells, and this is really where gas exchange occurs. So this is where oxygen enters into the circulation and carbon dioxide exits and then is exhaled. And this process of oxygen coming into the body and carbon dioxide leaving the body really just occurs through diffusion. And so there's this really large surface area of these alveolar sacs. In fact, if you take a 60-kilogram average male and sort of lay out the whole surface area of the alveolar sacs, you get about 2/3 of the surface area of a tennis court. So we have a very large surface area and a lot of reserve in the lungs, so that even if you have some damage, you still have quite a lot of function left. I also just want to point out the pleura here, which is the lining of the lungs, this is important when I talk later on. So really, I'm going to be talking about models from this more proximal region of the lung, the trachea and the bronchi, and these alveolar spaces. And what's important to recognize is that the SARS-CoV-2, the virus, can infect anywhere throughout the lung system. And there's actually a really elegant study from UNC showing that the ACE2 receptor expression, the primary receptor for the SARS-CoV-2, is most highly expressed actually in the nose and in the sinuses. And then there's a gradient of expression all the way down through the airway, so that you have less ACE2 the further down you go. But once we come to the alveolar sacs, there are type 1 and type 2 alveolar cells that we'll talk more about, and the type 2 alveolar cells, again, have high expression of ACE2. But it's important to recognize, and which is why we're modeling all of these different regions, is that the virus can really attack anywhere. So because of the different functions of the different regions of the lung, the histologies are obviously very different, and the cell types are very different. And it's these cell types that have these, really important functions. So when we're looking at the trachea and the bronchi, there's the pseudostratified epithelium. This is the mucociliary epithelium with mucous cells and ciliated cells. And then there are these submucosal glands, which are the primary site where most of the mucus is being made, which is then being cleared up and out of the airways. In the bronchioles, these are primarily just there to transport gases in and out of the body. And so they just have a columnar to cuboidal epithelium and then open out into these bronchoalveolar duct junctions and then into the alveolar spaces. And here are these alveolar spaces that I was showing you before. This is now a cross section, and it's nicely showing you the type 1 and the type 2 alveolar cells, or type 1 and type 2 pneumocytes. And what you can see with these type 1 pneumocytes is that they're these very elongated cells, and this is very important because they provide that very large surface area I was talking about before. And the type 1 pneumocytes contribute to what we call a respiratory membrane, which is just where these cells are very tightly opposed to the capillary endothelial cells across just a very thin basement membrane. And so as you can imagine, diffusion is very easy when this respiratory membrane is very, very thin. And then these type 2 alveolar cells are important because they're the stem cells of the airway, and they're capable of self-renewal and of differentiating into the type 1 pneumocytes. And it's these type 2 pneumocytes that tend to get injured, and they can respond with hyperplasia. And we see this a lot in a number of different lung diseases, including in SARS-CoV-2. So with that background in terms of understanding about the different regions of the lung and the different areas that can all get infected, I wanted to talk a little bit about what we're seeing in patients, because obviously this is primarily a respiratory disease, and primarily when patients get sick, it's a distal or parenchymal or alveolar disease. And what happens is that patients typically, not at the beginning of the illness when they more have fever or muscle aches or sort of that general flu-like symptoms, it takes on average, they say it's about a median of 8 days, but it's not right away, but it's a little later on, about a week later, that patients present with shortness of breath or dyspnea. And at that time, they may be found to have hypoxia, where the oxygen levels in the blood are low. And this is usually because of pneumonia, where there's inflammation in the alveolar sacs. And what we found is that about 80% of people will be fine and not have any overt symptoms of dyspnea and hypoxia, but it's around 20% of patients who will have some of these symptoms. Now, the numbers are really difficult because it seems like in every country and even within different hospital systems around the US, we're getting a bunch of different numbers. And this probably reflects patient populations, access to medical care, a whole lot of different issues, which is why it's hard to get these numbers. In some medical centers, they're very aggressive about ventilating, and so we see numbers even up to about 30% of these patients who get ventilated. Some medical centers don't really, are not very aggressive about intubating and ventilating, and that number might even be down to 2%. But I'd say on average, it's about 12 to 24% of patients who actually go on to require mechanical ventilation because of this pneumonia. And what's interesting is that if you just take inpatients in hospitals and you do a chest x-ray or a CT scan of their lungs, even if they don't have symptoms, you'll find that they have abnormalities. And this may be because some of these patients are younger and have a very large alveolar reserve, like we talked about earlier, because we have so many, we have billions of alveolar sacs. And then the other things that we can see in patients are more later on, which is this bronchiectasis, where there's dilation of the bronchioles with mucus accumulation and cellular debris accumulation. We can have a pleural effusion, which is basically water around the lung, pericardial effusion, which is water around the heart, and then the lymph nodes can be enlarged. And in this chest x-ray here, what I'm showing you with the arrows is that there are sort of these pale, sort of the whiter areas. So black will on the chest x-ray, the air will be seen as black, and the white is where these pneumonic infiltrates are. You can see a lot of them occur actually under the pleura, or the lining of the lung around the outside of the lung. And that's probably more clearly seen on the CT scan, where you can see these ground glass opacities, which are quite classic, often around the periphery, just under the pleura, and it's also seen over here on this side. And then there's actually some fibrotic changes that are starting to occur here as well. So this is just some of the findings. Obviously, there's been a range of different findings across different patients, but this is just some of the more typical findings. So really what these patients are experiencing when they end up needing mechanical ventilation is a disease called Acute Respiratory Distress Syndrome, or ARDS. And ARDS is kind of like a bucket diagnosis, and it occurs in a lot of different scenarios. So other viruses can cause ARDS in addition to things like sepsis or severe burns. A number of different scenarios can lead to respiratory failure with ARDS. And really what this is in the early stage, when the virus enters, there's obviously an immune response, and so the macrophages are activated, the T cells are activated. And then later on, when you get to the severe stage, what's happening now is that there's a lot of cytokines, the sort of cytokine storm that people talk about, lots of inflammation, IL-6, GM-CSF, a whole lot of cytokines are being produced, and there are these hyaline membranes that form. So this is a mixture of fibrin and edema and dead and dying cells and inflammatory cells. But as you can imagine, this hyaline membrane is quite thick. And so what happens is that this very thin respiratory membrane across which gas exchange is occurring is now very thick. And so it's really hard for oxygen to diffuse into the body and carbon dioxide to diffuse out. And this is where the hypoxia is occurring in these patients, is because of the hyaline membranes. And there's some controversy about whether the hypoxia seen in SARS-CoV-2 is more impressive than what you typically see in regular ARDS. And so people are talking about this atypical ARDS, but what really, there's no consensus on that right now, but this is really what we are seeing in patients, is the development of hyaline membranes. And so this is just an H&E section of a patient showing you this eosinophilic material, which is the hyaline membrane. And then you can see that there's lots of inflammation around this alveolar sac. And so of course, gas exchange will be really difficult. And this is, by contrast, is the normal airway, showing these beautiful open airway sacs with this very thin alveolar walls across which gas exchange is really easy to occur. And then with time, what happens is that you get this fibroproliferation, and so you get a whole lot of fibrotic tissue forming, and then of course gas exchange becomes even more difficult. And so this is typically what we're seeing within patients. And so coming back to our modeling, what we've really decided is that it's very important to model the distal airway, even though it's really difficult to grow type 1 and type 2 pneumocytes. But obviously, this is where the majority of the severe disease is. And so we need to model that. And in addition, we've decided that it's important to model the proximal airway because this is likely where the virus is replicating and then is the source of respiratory droplets which are infecting other patients. And I do really want to point out that everything that we're doing is in collaboration with Vaiti Ramagaswamy. And without Vaiti, we really wouldn't be able to do any of this modeling. So this collaboration is key because we're able to get live virus into these different models to study the disease. Okay, so the first model I want to share with you is this alveolar niche model. And this work was actually started a number of years ago in my lab by Dan Wilkinson, who was a graduate student from Bruce Dunn's lab, who tried to figure out how to recreate and really bioengineer a scaffold to replicate these interconnecting alveolar sacs that you see here in this micrograph from lung. And he's been helped by Luisa Meneses, who was an amazing undergraduate in the lab and now a technician in the lab and soon to be a graduate student at UCSF, and then more recently Chandani Sen, who's come in and taken over the model and is doing the SARS-CoV-2 modeling. And so the premise behind the generation of these interconnecting alveolar spaces and the scaffold for this is really quite simple, and it has to do with this the shape which is actually present in nature and even present in batteries, I found out, but it's called inverse opal geometry. And so the idea is if you're in a grocery store, if you could even get to a grocery store, but if you're in a grocery store and you think about a stack of oranges, so these are spheres, orange spheres, so these are the oranges, and if you imagine the spaces between the oranges, which is the green, if you could fill those up with cells and then remove the oranges, now you have these interconnecting alveolar sacs. And so this is really how Dan developed the bioengineered model. And I don't want to spend too much time on this, but really what he did is to create these alginate beads. Alginate's an inert material, so very easy to work with and not a problem with cells. He coated the beads with collagen. And then what I'm showing you is how he created this rotating bioreactor where the cells are then plated together with the collagen-coated beads. And when you do this and they move together, and what he'd done here is to hook up a GoPro camera. This is like the mounting for the GoPro camera, but I hope you can see, whoops, sorry, looks like I may have stopped sharing. Sorry, let's try again. Okay, hopefully this is working again now. You can hopefully see that the beads are moving along with the cells as they move through the rotating bioreactor. And then what happens is that the cells are able to coat the beads evenly. This is a calcine intravital dye in green, showing you that the beads are evenly coated across with cells. And then if you include cells, including fibroblasts, what happens is that the cells create these bead-bead bridges and pull together, creating these cell-coated beads that basically create these interconnecting air spaces. And so this is a model that we're actually using to study lung fibrosis. But as we realized what it really is it's a great co-culture method to get in a large number of different kinds of lung and airway cell types. So here I'm showing you fibroblasts with pimentin, pan-cytokeratin for different epithelial cells. This actually, on this side, is actually normal lung tissue, which is stained for pan-cytokeratin in green and for fibroblasts in pink. But you can see that we really are nicely able to recreate these interconnecting alveolar spaces. And we've grown endothelial cells as well in the mix, and we're hoping to start to introduce inflammatory cells like macrophages as well. And so with this as a co-culture system, we thought that it would be really useful. But one other thing that we realized is that the mechanobiology of the lung is very important, because just as you're sitting here and watching this, you're breathing and your lungs are stretching and releasing all the time. And that's a very important component of the niche. And so we teamed up with Jimin He and Mutian Hua, and we basically have been working with them to create a stretching system. And what we wanted is to create a system where we weren't just stretching from the outside, but actually each individual bead was stretching so that the cells on the beads would be stretched individually too. And what they have is this very cool bioreactive polymer called called NIPAM, where at 38 degrees, it's the material contracts, but at 26 degrees, and we've been using more like 33 degrees, there's expansion of the material. And so what Mutian did is to create, is to use this particular polymer coated with collagen and coated with cells, shown here again with the calcine intravital dye, and then create a microfluidic system where it could flow hot water and cold water very quickly around these organoids. And when he does that, what he's able to do is to stretch individual beads. And so I hope you can see in the movie how the beads are stretching, and so the cells around the beads are stretching and releasing and stretching and releasing, just like we would have with the mechanics of breathing. And while we haven't been using this for the SARS-CoV-2 infection, I think that there are some possibilities to use this modeling as well, especially seeing these cues of breathing are very important. Okay, so now I want to show you some of the work in progress of what we've been doing with this particular model and how the infection's been going. So one of the things that we realized up front is that a lot of the small airway epithelial cells that we were using in the model that I showed you previously were actually really resistant to infection. And so we've done a lot of iterations of this model now to try and figure out which cells will actually infect. And we think that we finally have figured that out. But this is just showing you with Vero cells, which are these monkey cells that have really no immunity to the virus and infect very easily. This is just a low power view of our organoids, and this is showing you the ACE2 staining of these cells around the beads. And you can see that the Vero cells here in green, we're able to detect the virus very easily within the Vero cells. And finally, in collaboration with Barry Stripp's group at Cedars, who are actually getting fresh lung tissue in and isolating the type 2 alveolar pneumocytes and then culturing them in Matrigel, we've been able to get those onto the beads as well. This is the mock infected. You can see that there's no virus. And now we're able to detect virus at a level where we can actually get to screen this particular organoid. And so that's exactly what we're setting up right now. And so because this bead system is completely scalable, where we can go anywhere from 25 cell-coded beads to thousands of cell-coded beads, we're able to scale up into a 384 well plate system and have very, very comparable organoids in each of the little wells. And so now what we're doing is gearing up to screen thousands of compounds in the little organoids, and we'll be able to tell the effective compound from the non-effective compound, and then be able to use Z-prime statistics to make sure that our model is good by looking at our controls of treated versus not treated, so with positive controls and negative controls. And so this is what we're doing right now, and really the workflow is going to be that we're going to pin compounds within these 384 well plates, then we'll plate the cell-coated beads, give about 3 days or so for the organoids to form, and then Vitae will infect the organoids. Then we plan to 2 days later fix and stain the organoids, and then use high throughput confocal imaging in the core, in the MSSR core, and then we're able to count the number of infected cells by the total number of cells and find out which compounds in this primary screen are able to reduce and or completely abrogate infection. And of course, all of this is being done with Robert Damaso in the MSSR core. And again, Robert is a key collaborator, because without Robert, we really wouldn't be able to run the screen. And we're super hopeful that we're going to be able to screen thousands of compounds with the goal first of using libraries of FDA approved compounds, because those are the ones that could be most quickly repurposed to the clinic. Okay, so that's all that I was going to say about the distal lung organoids. And now I wanted to move on to the proximal airway, the mucociliary epithelium. And we have more data here because we found it easier to infect these cells. And so basically the way this model works is this has actually been around for decades. And here we use airway basal stem cells, which are the stem cells of this proximal part of the lung. And when we what we do is we basically are able to get in human lung samples at the time of lung transplant, or we can purchase these basal cells. So we basically, when we get these samples in, we use enzymatic digestion and stripping of the airway, of this particular airway. And when we single cell dissociate the airway epithelium, we isolate out the basal cells and all the other cells die. And we can plate those basal cells onto a transwell membrane system. Here we're calling them human bronchial epithelial cells, but really they're these airway basal stem cells that survive. And when we put them onto this transwell membrane, we keep them submerged. So we have media in the top part of the transwell and the bottom part of the transwell. And we grow them for about 5 days or so. And what happens is a beautiful self-renewal of the basal stem cells, such that they form a lovely monolayer. They have tight gap junctions and they form this very good epithelial barrier. And then at that point, if we remove the media from the top of the transwell and expose the cells to air on their apical surface, but keep the media in the basal compartment, the cues to differentiate are there, and the polarity is there, such that these basal cells are able to differentiate into these mucus-producing cells and these ciliated cells. And so this is now a very nice model for us to study. And this work is being done in the lab by Arinima, Prakasya, Chandani Sen, and then Abdo. Dara has also been helping because what we've been doing is taking these cultures now and exposing them to cigarette smoke or to vaping aerosols. And this was a project that was ongoing in the lab before COVID-19. And is something now that we're using to explore the effect of the virus in addition to cigarette smoke and vaping. And so what I wanted to show you is just what...
Quick question. Can you take a question?
Yes, of course.
Okay. These primary HBECs that you diagram here, are they similar to the HB techs that you can purchase from a few different companies?
Yes, so these are available from a number of different companies, and they are exactly those primary airway cells. They actually can be frozen down and thawed and be salvaged quite readily. So they are available from a number of companies.
And they're essentially the basal stem cells that are able to replicate in culture.
Exactly, that's correct. So if you don't put them on, on the transwells and expose them to air, they'll just keep replicating as primary airway basal stem cells. They don't really differentiate. They do get some secretory genes that are turned on, but they really will not form the mucus and ciliated cells unless you give them the air liquid cues, interface cues.
But they express keratins and not collagens.
Exactly. They express keratin 5 most notably, and probably some keratin 14 as well.
Okay, thank you.
Okay, no problem. So what we do is we can actually immunostain in the transwell and then use confocal imaging. And this is an example showing you with acetylated beta tubulin what the cilia look like under the confocal. This is MUC5AC, which is a mucus stain. And you can see that there are mucus expressing cells within this culture as well. And obviously from patient to patient, we see some differences in terms of how many mucus cells there are and how many ciliated cells there are. And here's the keratin 5 staining of the basal cells. And the more differentiation there is, and the less there is in terms of injury repair, the fewer K5 cells we see, and the more we see of the differentiated cells of the mucus and the ciliated cells. So as I was saying, there's obviously a large number of things that we can do once we infect these cultures, but one of the big questions in the lab has been how does smoking and how does vaping influence the viral infection? Because this has been a big question which people have been asking because the results have been sort of mixed, and probably this is because the smoking histories of some of the patients have not been accurately recorded, so the demographic data has been a little bit controversial, but it seems to be quite clear now that patients who smoke are more likely to get severe disease and have a higher chance of, there's higher mortality in smokers with SARS-CoV-2. And so this is the experimental design of what we set up to study. This is just looking at cigarette smoke, not the vaping, but just the cigarette smoke, of how cigarette smoke influences the virus. And so what we do is we plate those human bronchial epithelial cells or airway basal stem cells on day 0, and we let them grow for about 4 or 5 days, and then take them to the air liquid interface. And then we let them grow for about 19 days or so. And then at that point, they're really nicely differentiated into all the different cell types. And then we have the smoking protocol where we use these research grade cigarettes and we expose the surface, the apical surface of these cultures to cigarette smoke for just 3 minutes a day, just once a day for 4 days. And then we give them a rest day, and then Vitae infects them in the BSL-3 high containment area, and we then wait for, sorry, it looks like my numbers here are off because we're actually waiting only 3 days. So this would be at day 32, and then we deactivate the virus by adding either methanol or a PFA, and then we're doing a whole lot of analysis on these cultures to try and understand directly what the influence of smoking is on viral infection. And so, and I should also say that this is being done by Arunima and Chandani, as well as Gus Garcia from Vitae's lab, who's a very talented technician. And so here's some of the preliminary data that I wanted to share with you. And so this first group here got no cigarette smoke and no virus. So this is a control group. And you can see that there's a large number of ciliated cells, and there's no viral infection, so we don't see any evidence of the virus. This is just cigarette smoke without virus, and you can see that there's a loss, and there's a big loss of the number of ciliated cells. And this is something that my lab has found from before, is that cigarette smoke is really good at removing the ciliated cells. But now here, if we look at just the viral infection, this is without cigarette smoke, you can see that there's a patch of infected cells here. And we do see this very patchy effect of the virus on the airway epithelium. And you can imagine that maybe one cell got infected and then infected the cells around it. And only about 10 to 15% of the airway epithelial cells will infect, which suggests that there's a fair amount of innate immunity within the other cells that are fighting off the virus. And you can see that really we don't see much in the way of loss of ciliated cells with the virus. But now, when you have cigarette smoke and the virus, we see a lot more viral infection. So here we see more patches, and the patches are larger. And so if we look now at so the same groups, and looking at now at mucus staining with MUC5AC, this is the controls with no cigarette smoke, no virus. Here is with cigarette smoke, and we see a big increase in the amount of mucus production, which we've seen before. And of course, we know that smokers produce a lot of mucus, so that makes a lot of sense. This is just the virus, and you can see, and we've done the quantification, actually there's no increase in the amount of mucus. There's no statistically significant increase in the amount of mucus from the virus, but you can see these patches of cells that are infected. And now with cigarette smoke and with virus, we see obviously these bigger patches. And again, there's actually no statistically significant increase in the number of mucus cells when we actually did count, despite the fact that smoking will increase the amount of mucus cells. And then Gus also looked at the viral load, and we did see that there was an increase in viral load with smoking. We suspect that it may even be higher than what we're seeing, but we think that some of the cells that are heavily infected are dead and dying, and so we're just not capturing them in the cultures. And then this was an interesting observation that we made that we're still really interested in, but haven't done enough on. But this was, we looked at proliferating cells in the cultures, because we've seen before that in the setting of cigarette smoke, this is without virus, that we see a fair amount of proliferation of basal cells. This is Ki67 and pCNA, so that we would get all proliferating cells. And so on the top row is the proliferating cells, on the bottom row is the keratin 5 basal cells. And so we do see an increase in the number of basal cells with smoking. But what we found with a virus is that this was virus infection with and without smoking, is that there was really no increase in the number of proliferating cells. There was a little bit more with the smoking and virus, but it wasn't significant. And definitely with virus alone, we just really didn't see that proliferation, which would be the repair response of the basal cells in response to injury. And same thing with the keratin 5 staining. And this is quantified here, where you can see in the setting of cigarette smoke, we have an increase in the number of cells that are proliferating, but not so in the setting of virus. And the same thing with the keratin 5 cells. And so what we think is happening is that the virus is actually reducing that repair response that the keratin 5 cells would usually have to repair the damaged airway epithelium. And we think that that's an interesting thing. And this is borne out as well in the single cell sequencing that we're still analyzing right now, but we see a downregulation of a lot of genes within the viral infected cells, and it seems to be dampening down this proliferative repair response. And then another response that we're seeing that is being repressed, very strongly repressed, is the innate immune response. And this has already been shown by other groups, but we saw the same thing. And one of the things that's down is the interferon response. And so, so in order to look at that, what we did is to look at these ALI cultures, and this time to add some compounds. So what we did is our regular protocol of creating the ALI, smoking for 4 days, and then giving a rest day. And then an hour before Vitae infected the cells, he added either interferon beta-1, remdesivir, or no drug at all. And this is what the data came out like. And so obviously these are our controls, and we don't see any virus. In red is cleaved caspase 3, so we're looking for apoptotic cells. And you can see with cigarette smoke, we do see some apoptosis. Obviously there's no virus, so there's no virus staining here. But in the setting of just virus alone, which you can see over here, we see that the interferon beta-1 completely abrogated the infection. There was almost no infected cells. There was definitely a reduction with remdesivir, but it was not as striking a phenomenon as we saw with the interferon beta. And then with the cigarette smoke, obviously we see a lot more infection, and we saw that remdesivir and interferon beta were both really good, but interferon beta was somewhat better at this. And so that's where we are right now with the data. Unfortunately, I don't have the single cell data. It's still being analyzed, and so I don't have anything to show you yet, but we're just overall, like I said, seeing this repression of a large number of gene signatures that are probably really key in the virus responding to infection and repairing. And
Brigitte,
so,
another question here.
sure.
I wonder if you can assay the interferon response on a cell-by-cell basis histologically by staining for IRF3, which will be nuclear if you've got interferon response, and cytoplasmic if you.
Yes, that's a great point, and that's something that we're planning on doing. We also think it would be good to do single cell sequencing in the setting of the treatments to try and get a sense of exactly what is being turned on in the cell and how the cell is responding as well. So those are other things that we've been thinking about. But definitely understanding more about the interferon response we think will be helpful, as well as other responses. Because unfortunately, interferon is not going to be, as well as it does in the dish, it's not going to be something that's going to be really tractable to give to patients because it makes patients so sick. So this is not going to be a therapy that we're going to be able to use. So having some other target would be better.
Right, but I was just mentioning as a way of assaying in the same experiment where you're doing the immunofluorescence.
Yes, no, absolutely. I think that's
which cells
a good point.
have activated interferon into screen.
Yes, absolutely. I think that's a really good point, and these are the kinds of things that we need to do, is to look more at the pathway.
Mm-hmm.
Okay, so so I guess I'm right near the end, but I just wanted to acknowledge that. Gosh, I think it takes even more than a village, maybe like a whole city of people and collaborators who've just helped us so much. I just want to acknowledge people in my lab. I think as I went through, I try to acknowledge people, and I apologize if I left anybody out, but the whole lab has been working really hard through the pandemic to try and do these studies despite, like, the crazy conditions. So they really are amazing. And then, like I said, without Vitae and without Robert, we really wouldn't be able to do this work. I also wanted to acknowledge Catherine and Justin, who are working on the single cell sequencing, and Barry Stripp's group, who in parallel have also been trying to work on different models. And we think now a hybrid model with their AT2 cells and our organoids will be the way that we can actually get to true high throughput sequencing. So we're really excited to do the next set of experiments and the actual screening. And then, of course, we're always really grateful for our funding support. We really luckily we were able to write a number of grants for specifically for COVID funding, and be able to get some funding in the lab, and that's been able to support these really expensive studies. So we're really grateful to the Broad and to CIRM and TRDRP, as well as the Mercator Center, and then to the NIH. So with that, I'm going to stop, and I'm happy to take any questions.
Great, thank you so much, Brigitte. That was great. I think we have 2 questions already from the group chat. I'll just read them out to you and you can respond. And then anyone else that is that would like to ask a questions after these 2 questions, feel free to show your face and ask directly to Brigitte. So the first question from Hui Hui Ye, does gender difference lead to different infection rate in this model? Male gender is associated with higher mortality. Thanks.
Yeah, that's a really great question, and something that we're really interested in. We've realized that the model can be used to study gender, age, ethnicities, and this is a direction that we're going in right now, because we can get cell types from all of these different kinds of patients. So unfortunately, I don't know the answer yet, but it's something that we're studying, and hopefully we'll be able to know that fairly soon. The trouble, I think, with patients is that there's so much heterogeneity that we're going to have to study a large number of patients. We have to do these studies on many, many patients to be able to come up with the final answer, but we hope to be able to get to that.
Cool, thank you, Brigitte. Okay, so the next question is from Dan Gershwin. Is there a difference in O2 versus CO2 gas exchange that would explain the lack of sense of hypoxia or lack of CO2 drive to breath in early stage patients who have substantial hypoxia measured by O2 saturation?
That's a really good clinical question, and unfortunately I'm not an ICU doc, so I don't know if I'm going to be able to answer that really well, but my understanding is that so I think that the CO2 is not as much of an issue with the diffusion as it is the oxygen, but unfortunately I don't know the physiology behind that, but I think that's something that an intensivist would be able to figure out, because it's very true that we that the major issue has been the hypoxia with these patients. But I don't know why the CO2 is not increasing that respiratory drive. Sorry, I can't answer that.
This is Gay.
Thank you. Yep.
Sorry. That was a fantastic presentation and an amazing model, and really so many interesting questions that it produces. Just a really small thing is, why did you choose interferon beta 1 for those experiments?
That's a good question. So some of it is just that something we had on hand, and something that Vite had been playing with before with other experiments. But some of it is because of the single cell sequencing, where we did see that the interferon response was downregulated and strongly across all different cell types. So we thought it might be different with insulated cells or mucous cells, but it's not. All the cell types have this downregulation of the interferon response genes. So we thought it would be interesting. And of course, there's also other publications showing that the interferon response is important in a number of different settings in the literature.
But it was interferon beta 1 specifically that you...
This is the one that we tried. We haven't tried any other interferons, just the beta 1.
Now, why would it make any difference whether it was interferon alpha or interferon beta or whichever interferon beta? Because they all bind the same receptor.
Yep, I don't know. Yep.
Okay.
But maybe
interferon gamma, that would be different. But I've heard of these differences between alpha and beta, and maybe in a physiological model, it has to do with how stable they are after they're given or something like that. But from my understanding, they activate the same receptor.
Yeah, no, we don't know. We don't have any experience with testing other than beta 1, and we've just done it once. We're gearing up to do it again.
Sorry, interferon beta and different interferon alphas, which are several, have actually different binding to interferon alpha receptor 1 and interferon alpha receptor 2. So beta, is very strongly favors the alpha receptor 1. Sorry. Yeah.
Okay, thank you. Yes, definitely. We would love help in terms of thinking this through.
All right, Brigitte, there's I'll jump in. There's a question from Jason Hyman. The primary output of the drug screen appears to be blocking cellular infection. Are there other mechanisms, for example, downstream inflammation, that can be measured in this screen?
Yeah, that's a great question. So in this screen, because we're probably going to just have epithelial cells, AT2 cells, and fibroblasts, just a few fibroblasts in there as sort of as supportive, and probably, going to be secreting growth hormones or growth factors in there. We don't have any inflammatory cells in this particular model. It would be super interesting, and something that we really want to do is to put something like macrophages into the model and look at the response, but it's not something we've done yet. And so for the purpose of the screen, especially keeping the screen really simple and easy, we decided to just go with infected cells versus non-infected cells. And that actually doesn't even take into account which part of the viral life cycle will be hitting with each of these compounds either. And that's something that would have to be teased out later on. So some of it might be related to viral entry, but some of it could be replication. We don't know yet. But this is just for the primary screen, and then after that there'll be secondary screens. I'm imagining several secondary screens, and then probably some animal studies before we could actually get it to patients. I hope that answers the question.
In a non-screen setup though, just as a sort of an experimental question, can you imagine that this could be combined with different sort of cell types, immune cell types, to see whether you could prevent infection with, let's say, iNKTs or something like that?
Absolutely, that's something that we really want to do, and we're collaborating with some groups, thinking about that.
All right, Brigitte, can I jump in and ask a question?
Sure.
So I thought when you talked about the ARDS, scenario, the hyaline membrane is really interesting. So I was wondering what your thoughts on in terms of macrophage infection by SARS-CoV-2, because we know for SARS, it does infect macrophages in the lungs. Do you think that the recruitment of the macrophages to this particular site and all the inflammatory signals are actually promoting infection of these cells that maybe normally wouldn't be infected as efficiently? Yes,
definitely. So I think what's happening, and I don't know, this is sort of like half like sort of like maybe a little bit of hand waving here, but what I think is happening is that when the virus infects, it's actually reducing the immune system within the infected cells, but the body is perceiving that there's infection, and so what's happening is there's this immune dysregulation, and so the non-infected cells are coming in and trying to clear the infected cells, and in so doing, producing the cytokine storm as a way to try to clear the infected cells. And then all of that dead and dying cells and the debris and the subsequent edema and then the fibrin that's produced, all of that is creating those hyaline membranes. But in the patients who don't go on and develop the hyaline membranes, but who clearly have infection in their alveoli, right? And this is the majority of patients, especially the younger patients and the patients who don't have the risk factors. They are obviously able to clear that infection without that immune dysregulation, right, of that cytokine storm. And so I don't know why there's that dysregulation and sort of that immune response, that sort of that over-exaggerated immune response in some patients and not in others. And I think that's probably the key, right, in terms of preventing severe illness, and how the risk factors put you more at risk there. I don't know. I think one of the things is that smoking, from our work, would suggest that you actually have the immune system. So we know that smoking will reduce interferon signaling and a whole bunch of other things that smoking is doing to the cells. So what I feel like is that when cells are exposed to smoking, their immune defenses are already down. So the virus gets in really easily. And then once it's in the cell, it's downregulating even more any kind of immune response. And so that may be causing some dysregulation there too. But I don't know for sure. I think it's one of the most important questions.
Great, thank you. And I think now we have two more questions. One from Melissa Spencer. Fantastic talk, Brigitte. Do you think the immune dysregulation could be due to reduced MHC class 1 or 2 levels and reduced antigen presentation?
Yeah, and I think it's possible, but yeah, I have no idea, but it's definitely possible. You know, on the clinical side, I take care of patients who have hemophagocytic lymphohistiocytosis, and these are patients whose NK cells don't work. And what we see in those patients is that when they can't clear virally infected cells, the classic culprit being EBV, the rest of the immune system gets really an over-exaggerated response, and they have cytokine storm, and you know they have a similar kind of effect as we're seeing with these patients. And so I think it could be almost anything that can cause that cytokine storm that causes an immune dysregulation, and it may be different in different people. So I'm wondering if maybe you can't clear the virus for some reason, which could be an NK cell problem, or could be, you know, some other cell problem, that maybe that's why you have this exaggerated response. But I'm really not an immunologist, so I shouldn't even be talking about this. I should just stick to the lung. But I am really interested in this, and if there are immunologists out there who would like to collaborate to try and answer some of these questions, we would be really excited to collaborate with people on trying to answer these specific questions, because I think in this model, we could start to address some of those things.
The next question is from Lori Shaker Irwin. Are you planning to create models for simulating vaping, marijuana smoking, CBD oil inhalation, etc., which may have implications for younger populations?
Yes, so I didn't present any data, but we have figured out how to, basically we're using the Joule pod, and we're able to take Joule aerosols and vape these ALI cultures as well. The data was not as clear in terms of the infection with the virus, but we do think that there's increased viral load with vaping, and we're actively working on that right now. The trouble with the vaping story is that there are thousands of different products, and each of the flavors is a different chemical, and each of the chemicals seems to have different toxicity in our liquid interface models. So we've studied several of these flavors now, like 5 different flavors, and we found that each one either caused more proliferation of basal cells, or worse loss of ciliated cells, or created more mucus. And so because each flavor is doing something different, then we feel like that flavor could influence a viral infection too. And how do you study thousands of these? So that I think is a real challenge for the field, is how do you study each all of the different chemical combinations that are in these thousands of vaping products right now. But I think it's a really important question, and I do think it's going to have a bearing on viral infection.
Great. The next question is from Manesh Paul. Great talk. As per the ACE2 expression, it's more in non-differentiated cells, especially in the ciliated cells, as compared to stem cells. Why in cigarette smoke, where the ciliated cells are reduced, the virus is infecting more?
So the ACE2 is tricky, because there's a fair amount of ACE2 expression, but not every ACE2 expressing cell is getting infected. And there's probably a number of different reasons for this. A lot of it probably speaks for just the innate immune response within cells. So we don't really know why one specific cell tends to be infected. And like I said, it's only about 10 to 15% of the cells within the actual liquid interface cultures that are getting infected. But what I think is, because we're smoking ahead of time, we've reduced the innate immune response in the cells in the liquid interface cultures. And so it's much easier for the virus to get in to more cells. And once the virus is in, those clusters seem larger. So it seems like it's able to infect more bystander cells as well. And so that I think is why we're seeing more infection with the smoking. And that seems like that's the most likely explanation. I don't think it's ACE2 related. Now, there is some data suggesting that ACE2 is upregulated by smoking. And we actually didn't see that. It's not statistically significant in our cultures, or we thought we saw an increase, but it didn't end up being statistically significant. And then it's thought that viral entry will actually reduce the ACE2 expression on cells, but that again wasn't statistically significant in this model. So I don't know if that's different to what's happening in the in vivo situation, or whether our ACE2 is representative or not. But ACE2 has been a little bit tricky to try and figure out.
And also, Brigitte, I thought it was interesting with the staining in your model. It's very clustered.
Yes.
which we don't always do for other virus infections. So I was also wondering about maybe like maybe there's more cell-to-cell spread versus free virus infection for these cells. So.
Yeah, I think you're right. It's really interesting how the infection is already clustered in specific areas, and yeah, I feel like I said, when we were starting to do these infections in the organoid model, we just didn't see a lot of infection. In fact, there was almost no infection, which was really surprising to us, that these primary cells are actually quite resistant to this virus that's creating so much havoc around the world, right? And so I don't know if it's just that our models are not representative, or whether this really is representative of most of the population, where you do just get a small amount of infection, just enough to spread it to somebody who's susceptible, who then, like those smokers, will get a really bad infection. And so maybe that does explain some of the heterogeneity that we're seeing across cases across the world, where if you're young and healthy, you mostly fight it off, but if you're susceptible, you get really bad disease. It's also interesting that we saw a fair amount of apoptosis, not in infected cells, but in surrounding cells. And we don't quite know what to make of that yet, but it's possible that the virus is having these sort of neighboring effects on the tissue that could also give you more severe disease, because you have more damage to the tissue and maybe your repair response is reduced.
Very interesting. Okay, one more question from Quan Chen. Can you elaborate on how you would co-culture immune cells like macrophages in either the organic or the ALI system? Do you imagine doing it with alveolar macrophages or monocyte-derived organoid?
Yes. Organoids, yes. So I think the easiest way to do this in terms of co-culture systems is to put this into the organoid model, because when we put it in the bioreactor, the spinning bioreactor, we can add any cells in. And so that would be what I would think would make the most sense initially, would be to put the macrophages into the organoid model. And that's where the majority of the macrophages are going to be, would be in the distal, alveolar spaces. The question of whether to do the alveolar, macrophages versus the recruited macrophages from bone marrow is a really interesting one. I don't know the answer to that, but probably we would have to try both. And maybe even the two of them in combination, because they are very different. The alveolar macrophages are patterned during development, and they're there and they really stick around there. That's where they stay, and that's how they are fighting infections locally. Whereas the others have been recruited from the bone marrow, and so probably have some different responses. But it would be interesting to test both, I think.
Great. I think that's all the questions in the chat room. Anyone else has a burning question? Great. Well, thank you so much, Brigitte. I learned so much. It was really interesting.
Thanks, everybody.
Yeah.
Thank you so much for the input, everybody.
Awesome. And I think next week we have Alex Hoffman.
Great.
Awesome. Look forward to seeing everyone next Wednesday, and have a good rest of the week.